Alright, so we've gone through phagocytes, we did mast cells, we did natural killer cells. Next we'll do inflammation. So with inflammation, there's 4 signs of inflammation: redness, heat, swelling, pain. These all have to do with extra blood being delivered, vasodilation. So when you dilate a blood vessel, if it gets closer to the surface of the skin, that's going to be the redness. Heat, remember that blood is a conduit of heat. So if you vasodilate, you're going to release heat too. The swelling, when you vasodilate, those blood vessels become permeable and fluid will leave. So that's where your swelling comes from. Another name for that swelling is edema. And then pain, whenever you have a bunch of fluid in that interstitial space that's normally within a blood vessel, it's going to push on nociceptors. And these are your pain receptors. So if these get activated because the fluid or the edema, the swelling is pushing on them, then it's going to cause a pain response in the body. Alright, so the inflammatory mechanism, again, I'm going to read through this, explain it through words, then I'll draw some pictures, annotate them, and we'll go from there. So with the inflammatory mechanism, again, nothing happens without a signal, right? So our signal is that the macrophage recognizes this pathogen, this foreign pathogen. And these receptors on the macrophages are called toll-like receptors. So our toll-like receptor is going to interact with the pathogen, and that's going to cause cytokines to be released from this macrophage. All right, cytokines are going to cause inflammatory mediator chemicals to be released by these things called inflammatory mediator cells. Alright, and then the mixture of the cytokines and the inflammatory mediator chemicals is going to cause vasodilation. So vasodilation is the opening up of these blood vessels, and that's going to cause hyperemia to occur. Hyperemia is just, emia refers to blood, hyper means lots, so it's basically more blood flow to this area. Whenever you dilate a blood vessel, this blood vessel will become permeable. So if it becomes permeable, then we know things can pass through it, like fluids and cells. So we're going to have exudate leaving. Exudate is this mixture of fluid and cells, and that's going to cause our edema that we just talked about. So if we go back to our signs of inflammation and try to connect some dots. So we had that vasodilation, which means hyperemia is coming. So the vasodilation caused the redness and the heat. And then because they're permeable, we could have that exudate leave, and that exudate, that fluid is going to swell our interstitial space, causing edema or swelling. And then again, that fluid is going to push on and activate these nociceptors or these pain receptors, which will give us a pain response. So if you want a little picture, this green is our skin. So we'll just say this has been damaged and we have a pathogen that's coming. So our toll-like receptor is going to interact with this pathogen, and this is going to release cytokines. And then I'll just have some other cells. These will all be our other inflammatory mediator cells, and these will release inflammatory mediator chemicals. Okay, so it's the interaction of these cytokines and inflammatory mediator chemicals that are going to cause our vasodilation. So let's draw a dilated blood vessel. So if we have this now, our dilated blood vessel, let's get us some skin. There's our macrophage and our pathogen. Okay. So we know whenever we dilate a blood vessel, it becomes permeable. So I'm just going to show a few slits there. And that vasodilation is also going to allow hyperemia, so we get this big rush of blood coming in, bringing in some other cells too. And because this blood vessel is permeable, this fluid can leave. So what's leaving is called exudate. Exudate is leaving the blood vessel, causing swelling or edema. Because if I drew some fluid in here, here's a bunch of fluid, here's a bunch of fluid, here's a bunch of fluid, and let's say this is just a nociceptor. It's going to push on that and activate a pain signal in our brain. So let's walk through that one more time. Toll-like receptors on our macrophages will interact with the pathogen, and that's going to cause the release of cytokines, which will cause the release of these inflammatory mediator chemicals. The cytokines plus the inflammatory mediator chemicals are going to result in vasodilation, which means that this blood vessel is going to become permeable, which means fluid can leave, exudate can leave, which is going to cause the edema, which is going to cause the pain. So that's the overall view. Now there's a couple steps that we want to include here. So if this is our blood vessel, our dilated blood vessel, okay, and this was our skin, we could either say that the skin just got punctured, or maybe this was a dirty nail or something, and it made a little hole through our blood vessel too. Regardless, either one of these, let me get rid of this, either one of these is going to cause our endothelial cells, which is this lining of the blood vessel, to release leukocytosis inducing factors. So this doesn't say leukopoiesis, so this isn't the production of red blood cells. Leukocytosis is just, osis is condition, cyto means cell, leuko is white. So it's a condition of having a lot of white blood cells, which just means that we're going to get white blood cells to this area. Okay, so these leukocytosis inducing factors are going to cause the release of neutrophils from our red bone marrow. Remember, red bone marrow is where leukopoiesis and erythropoiesis is happening. So at this site, we're going to get a release of neutrophils, which are these white blood cells, which do diapedesis and they do phagocytosis. So we already talked about those in the earlier video. Alright, so we have this vasodilation going with a bunch of hyperemia, which means more blood flow. And within this extra blood that's coming, we have a lot of neutrophils. Now, normally when you dilate a blood vessel, we think that it's going to increase blood flow, but what I want you to think of this as a garden hose. So let's say this is my hose, and I have fluid coming out. If I put a huge cut in the side of my garden hose, so there's a big hole, I'm going to be able to have fluid coming out, which means the fluid that is coming out at this point is not as fast. So what this does whenever, let me draw another picture. When I have this dilated blood vessel, this fluid that's coming in, this blood that's coming in, it's going to slow locally. And let's think why that might happen. If we have a neutrophil coming in and it's going super, super fast, it might miss its chance to diapedese to then work on, this is just a quick picture. Here's our this is our macrophage and our pathogen or whatever. So it might go too fast. So this slows locally to give these things time to be able to diapedese. So let me draw one more picture. Alright, so we have our blood coming in with these neutrophils. And again, remember, this is our endothelial layer, our inner lining of a blood vessel. So this endothelial layer is going to release something called a selectin. Alright, so selectin functions as an adhesion molecule. To adhere to something just means to bind. So let's put some selectins on the walls of these blood vessels. So if you had to guess, what do you think that these selectins are going to bind to? Probably these neutrophils that are being delivered through hyperemia, right? So here's one more picture. I have a bunch of selectins. And now I have these neutrophils binding and accumulating on the walls of my blood vessels. This process of selectin and neutrophils binding to the walls of blood vessels, this is called margination. So it's just an adherence, but it's a special term given to this kind of adherence, where you have adherence of selectin, neutrophil, and on the wall of the blood vessel. That's called margination. And so then these neutrophils can then diapedese, which just means to go across a blood vessel into the interstitial space where they can work on those pathogens that we were talking about. So now we're getting an accumulation of neutrophils. Remember, these neutrophils are part of what we called exudate. So exudate was that fluid plus cells. These are some of the cells. Following those neutrophils are going to be monocytes. And quiz yourself when a monocyte does diapedesis. Let me put one right here. If my monocyte does diapedesis across this blood vessel, what's it called now? a macrophage. So this whole process started with macrophages and is ending with the delivery of more macrophages. So these neutrophils and macrophages can clean up whatever's trying to get in our body through phagocytosis. So now we've connected some of our cells, some of their mechanisms of phagocytosis, along with this whole mechanism of inflammation. Inflammation isn't going to be the only place. Inflammation won't be the only place where we see phagocytosis. This is just one example of how phagocytosis is acting in another bodily process. Alright, so we've knocked out inflammation. Let's move to the antimicrobial proteins next. Alright, so antimicrobial proteins. We talked about two classes. We talked about interferons and I'm going to abbreviate those as INFs. And the other class is called the complement proteins. And we'll call these CPs for now. So let's do interferons first. This is the interferon mechanism. I'm going to write it out really quickly and then we'll draw some pictures. Alright, so the interferon mechanism. So a cell is going to get infected by a virus and that cell's going to die. But that cell, in an effort to protect its neighboring cells, is going to release interferons. So interferons are going to diffuse to nearby cells. Interferons are going to cause the production... This diffuses. Interferons are going to cause the production of a protein called PKR, and PKR is going to block viral replication. So another thing interferons do are going to activate macrophages, which will do that phagocytosis, and the natural killer cells, which are going to release those perforins, which we talked about in the last video. So if I want to draw a picture of this, let me draw a few cells. Actually, I'm going to do this a little bit bigger. And we'll say this first cell is going to be infected. We can't do dots, so these lines are representing a virus. Okay. So this cell is going to be infected, and it's going to release interferons in an effort to save the rest of its neighbors. Interferons going out that way, interferons going out that way, interferons going out that way. Okay, and it's going to diffuse into here. I'm just going to do an eye just to show that these cells have absorbed some interferon. And now these cells can start making PKR. So eventually this virally infected cell is going to lyse, and it's going to release a ton more viruses. So here's a bunch of viruses. And these are eventually going to diffuse into these cells. But the PKR is going to block this synthesis. So basically the PKR will say you can't replicate in here. So it's going to save it. So the way I remember which cell is the one releasing IFNs, I always think of, that cell's going to die. The way I remember that is IFN, pardon my language, but I think of this cell says, I'm fucked now. So in an effort to save all the other cells, it will release interferons, which can diffuse into these neighboring cells, causing the stimulus of PKR, which will stop that viral replication process. Cool, so that's the interferon mechanism. Again, virally infected cell is going to release interferons, diffuse the neighbor cells, PKR protein synthesis stimulated, and it's going to block viral replication. So in the steps, step 1, virally infected cell releases interferons. These interferons can then diffuse out. It's going to stimulate PKR, and it will stop the viral replication process. So next we're going to do the complement proteins. So these are blood plasma proteins. They're always circulating the blood. They're not always active, but they're going to help destroy some pathogens. So there's two systems. We're going to say the complement system mechanism 1, and we refer to this as the classical. and then complement system mechanism 2, and this is the alternative. Okay, so what's the difference in these? The classical system is going to, this complement protein is going to bind to an antibody, which is going to be bound to a pathogen. So it's going to bind to an antibody-pathogen complex, while here in the alternative mechanism, the complement protein is going to bind directly to the pathogen. So let's draw two pathogens. Here's a pathogen, here's a pathogen. And the first complement protein, it's going to have an antibody. Okay, cool. And the second one, we're just going to put a complement protein. So this would look like this for the first mechanism. Pathogen, antibody, complex, and then the complement protein attached. The second one is just a complement protein attached to the pathogen. So either one of these systems is going to result in lysis, phagocytosis, or inflammation. And we've talked about all those in previous videos and previous examples. So either one of these, regardless of whether it's the first, the classical, or the second system, the alternative, it's going to cause lysis, phagocytosis, or inflammation. So this might cause, this complement protein might have a binding site. Let's just do that right there. And it might attract a macrophage. So let's just say this is a macrophage. And that would cause this to be phagocytized, bringing it in, extending its plasma membrane into that phagosome, using lysosomal enzymes to break it down. This second one, I just want to do a quick example. If we were seeing that alternative mechanism, complement proteins can cause lysis just by themselves. So let's say this is my surface of my pathogen. Well, if I have a complement protein, here's my complement protein bound to it. I want you to think of everything within that circle as being inside the cell. So this is just, this is a big hole now. This is basically punching a hole through the plasma membrane, but instead this is a complement bound to it, just making a hole. So now everything from inside that cell can come out, and this cell will die. Remember, if you're losing your cytoplasm and losing your organelles, that cell's going to die. So this is the alternative system where you're seeing a complement protein do lysis directly. Alright, so that's our antimicrobial proteins. I don't think that I've said, I might have said at some point that an antibody was an antimicrobial protein. If I did, I did not mean that. Antimicrobial proteins, again, consist of those interferons and complement proteins. Alright, our last one is fever, and remember, this is our only systemic response. So systemic means that it's going to happen all over the body. This inflammation we saw, that's only happening at that very localized region. You're not going to get that huge vasodilation all over the body, or else you get a huge drop in blood pressure, which isn't good. So systemic, this is going to cause an increase in body temperature across the whole body. So let's look at the fever mechanism. Alright, so what is a fever? It's an increase in body temperature. And let's see the mechanism behind it. So leukocytes, those are our white blood cells, and macrophages, which are a type of white blood cell. So I don't know what he meant by that, but just think of leukocytes and macrophages if you see them in the question SEM. And these two things, whenever they interact with the pathogen, are going to release something called a pyrogen. What's a pyrogen? It's a chemical, and what is pyro? Pyro has to do with fire, okay? So this is a fire chemical. What that means is it's a chemical that's going to cause an increase in body temperature. So these pyrogens are going to go to the hypothalamus. Remember, the hypothalamus is going to send signals to our medulla oblongata to help change automatic body responses. So this hypothalamus, in this case, is going to cause our thermostat to change. So hypothalamus is going to increase our set point of our body temperature, which is normally at 98.6, somewhere around there, or 37 degrees Celsius. And it's going to go, let's just call it 100. All right, so this is a systemic increase in body temperature. So what's going to happen? Some things, you're going to sweat. You're going to shiver. when you sweat, you're going to lose things like zinc and iron, and these things are used by bacteria to reproduce. So, and then as you're shivering, remember shivering is that involuntary skeletal muscle contraction. That's going to cause you to sweat more, release more zinc and iron so that these bacteria can't replicate. Shivering is going to that's going to be your mechanism, one of your mechanisms to increase body heat. So it's not like your body just automatically says, okay, I'm at 100 degrees. It has to do something to help get you to that temperature. So the shivering is going to cause that increase in body temperature, because whenever you contract a muscle, you produce heat. So this is super, super fast, rapid muscle contractions that are going to cause a lot of heat in the body. Why do we want a lot of heat in the body? Because viruses and bacteria only have a certain condition where they can function and live and replicate. And say a bacteria gets in our body and says, man, this is the perfect conditions for me to replicate. Well, if you increase that body temperature, it can't replicate, it might die, it might break down. You know that if you increase temperature to a certain point, you can get denaturation of proteins, so it might destroy some of the proteins that virus or the bacteria was using to replicate. So that's a fever. Systemic body temperature increase through shivering, by changing the body set points through these pyrogens. The pyrogens released by leukocytes and macrophages, they're going to go to the hypothalamus to change our set point to increase our body temperature, and I just gave an example of 100. Okay, so those are all our mechanisms. So a quick little recap. The skin, remember, it's a big barrier, so it's just going to cause, it's a bunch of barriers that a pathogen would have to get through to get into our body. These mucous membranes can trap things. They mix with acidic secretions. Then it can help break down viruses and bacteria to destroy them. Then we went into our internal. We did phagocytosis, where we recognized a pathogen, pulled it in through this phagosome, broke it down with some lysosomal enzymes. Mast cells, we said it was quote-unquote phagocytosis. We didn't get into the physiology, but we do know that they have a bunch of histamine, which is going to be a vasodilator, we'll see later on. Natural killer cells are these T lymphocytes that are part of our non-specific immune system. Normally we think of T lymphocytes as being just for the specific immune system, but these are T lymphocytes that have multiple receptors, so they can attack multiple things. Inflammation, we saw a mixture of phagocytosis eventually through these macrophages and neutrophils, but we saw the macrophages causing vasodilation, a bunch of release of, or a bunch of extra blood flow to this site. Vasodilation is going to cause permeability changes in our blood vessel. It's going to allow things to diapedese, that exudate the fluid in the cell is going to cause the swelling. So again, inflammation, we're going to have the redness from the hyperemia, the heat from the hyperemia, the swelling from the edema. Swelling and edema are the same thing, from this exudate leaving the vessels, and the pain from the swelling pushing against these nociceptors. Then we went into these antimicrobial proteins, the interferons being released by a virally infected cell to save its neighbors, and the complement proteins, which has the two mechanisms, the classical, which is going to work with antibodies and the pathogen complex, or just the alternative, where it's just a complement protein, and that's going to either of those sets of mechanisms is going to result in phagocytosis, lysis, or inflammation. And then we saw our systemic